Hi, everyone. And now we are talking about muscular tissue. So we just talked about muscles, fascicles, and the muscular system lecture, and we're going to move on to muscle tissue. So we know that each muscle is made up of fascicles, which are made up of muscle fibers, which are made up of myofibrils, which are made up of myofilaments organized into sarcomeres. Sarcomeres are our contractile units, and we will spend a lot of time talking about muscle contraction in this lecture. But first, we are going to talk about this comparison table. You will be asked a lot of questions about this on the quiz, differentiating between skeletal, cardiac, and smooth muscles. This is an old muscle tissue comparison table, but you can read through this. We will go over it as well. But basically, we have three different types of muscle, right? We have skeletal, we have cardiac, and we have smooth. So on the microscopic level, skeletal muscle is characterized by long cylindrical tubes, very parallel, not branching, striated. Anytime you see striated, that just means stripy. So a bunch of stripes. Cardiac muscle is branched and it has intercalated discs at the end of these branches, which we will definitely talk about more when we talk cardiovascular in Anatomy 2, in Anatomy and Physiology 2 next semester. Where cardiac muscle and smooth muscle have a single nucleus, skeletal muscle is it has many nuclei, so it's multinucleated. Let's see what else we got here. Smooth muscle, it tapers down at the ends. It's pretty thin. It's a different shape than everything else, so that kind of differentiates smooth from the rest as well. Should be pretty easy to identify skeletal, cardiac, and smooth muscle if we were looking at them through microscopes. Location, skeletal muscle is mostly attached by tendons to bone. Cardiac is only found in the heart, and then smooth muscle is found in a lot of different organs, digestive system, skin, blood vessels, all kinds of places that aren't attached by tendons to the bone, or, isn't the heart, is probably going to be smooth muscle. Contractile proteins. So now we're over here. Contractile protein organized into sarcomeres. So we just talked about what sarcomeres are, or glanced at it anyway. We're going to talk about a whole lot more here soon. But skeletal muscle and cardiac muscle are organized into sarcomeres. Smooth muscle is not. Sarcoplasmic reticulum. So you remember the endoplasmic reticulum, the ER. So sarcoplasmic reticulum is basically like ER for muscle cells. Sarco anything is going to be muscle tissue. So we have a lot of sarcoplasmic reticulum in skeletal muscles. We only have some in cardiac, and we have very, very little in smooth. Transverse tubule, so this has to do with contractile patterns. It's found in skeletal and cardiac muscle. Where are we? Yep, but not in smooth muscle. So cardiac and skeletal, but not in smooth. Junctions between fibers, there aren't any in skeletal. Cardiac we'll talk about when we get to cardiovascular and AMP2. And then remember those tight junctions, gap junctions, desmosomes we talked about in our earlier lectures? Here's pretty much where they come into play. So gap junctions, desmosomes, and then gap junctions and visceral smooth muscle here. Autorhythmicity. Okay, so does it make its own rhythm? Skeletal muscle does not. Skeletal muscle is completely voluntary, so it is not automatically making its own rhythm. Cardiac muscle does. It's involuntary, right? We can't control how our heart beats. So it has its own autorhythmicity. Smooth muscle has its own rhythm. You don't tell your intestines whether or not to move fecal matter around, right? It does it on its own. They do it by themselves without anything voluntary happening. Let's see. Regulatory proteins for contraction. For skeletal and cardiac, we have troponin and tropomyosin. And then for smooth, we have something a little different called calmodulin and myosin light chain kinase, which we are not going to talk about here. And then nervous control. So with skeletal muscle, it is very voluntary. That's our somatic nervous system. Whereas the other two are involuntary. They are our autonomic nervous systems. And then contractile regulation. It's going to be the same in cardiac and smooth muscle, but it's going to be different in skeletal muscle. This is pretty much what we just talked about. It's a summary of the previous slide, but this is for the folks that prefer lists rather than tables. So here you have a picture of skeletal muscle. You can see how it's arranged in parallel lines with striations, multinucleated, a bunch of nucleuses together. Cardiac muscle, however, a little different. You can see the branching, you can see the intercalated discs, single nuclei in each. And then smooth muscle, it's a little bit different from the rest with its shape, tapered down both ends. And yeah, this pretty much just organizes everything that we just talked about. Yeah, voluntary. This one's not autorhythmic. Okay, cool. Autorhythmic. Okay, moving on. Take a moment to watch these two videos. The first one with Amoeba Sisters is 8 minutes long. It'll give a little bit of a refresher over the previous chapter and go over muscle contraction, but both go over muscle contraction. This one's a little bit more involved. It's a little bit longer. And then we have a quick little McGraw-Hill animation. This animation is 30 seconds long. So this is going to be a quick animation over what we're going to talk about. So watch that. It is embedded into this PowerPoint. So this is about how muscles are contracting, and this is what we're going to talk about. Okay. Striation anatomy. So striations give muscle that striped appearance. If the striations are close together, then the muscle has contracted. If the striations are far apart, the muscle is relaxed. Sarcomere. Sarcomere is the smallest functional unit of skeletal muscle from end to end. Each sarcomere is its own contraction unit. So if you put a bunch end to end to end, then you create the whole muscle. Just a few of them contract, then there's going to be a little muscle contraction. But if you have a whole bunch and a lot of them contract, then you have a whole lot of muscle shortening and a whole lot of contraction. The sarcomere is just the smallest possible unit of said contraction. The sarcomere is made up of thick filaments and thin filaments. So thick filaments are mostly myosin, and then thin filaments are actin with some other proteins called troponin and tropomyosin. We just mentioned those, and we will talk about those a whole lot more in an upcoming slide. There is a Z line, so that makes an end of a sarcomere, and it's in a Z shape. From Z line to Z line is one sarcomere. They're also the proteins that connect the actins together, the Z lines are. And then we have an M line. You're going to see that here in a second too. And those are proteins that connect all the myosins or the thick filaments together. Okay. So this is just a schematic of a sarcomere. So we're going to say that this is one sarcomere unit. This side right here is the Z line. But if they were clicked together, then you would just see a Z forming all the way down. It'll look like this. Okay. So this is one sarcomere. But anyways, okay, we're just going to go with the schematic here for now. So anyway, from one Z to the other Z is one sarcomere. This right here is a thin filament called the actin. Actin. The Z line connects the actins. This thick line in the middle is a thick filament called myosin. These thin lines, the actins, are our thin filaments. Thin filaments are connected by the Z line. Thick filament is connected by this M line. So the M line will go all the way down and connect all the thick filaments together. So what that looks like, these dark purple thick bands are our thick filaments, and this purple crossbeam is our M line. So you see how that connects them all. Okay. So thick is mostly myosin, so think M line, myosin, connects the thick filaments. Z line connects the thin filaments, the actins. So it keeps them appropriately spaced. It prevents them from spreading too far apart or scrunching too close together. Z line spaces it out as far as it's supposed to go, and it stays that way. Okay. Let's see. Okay. H is just that little space, that little gap between the M line and the thin filament. So that's the Z line we already talked about. The I band, it gives you a light stripe. So when you look at a sarcomere, you see a bunch of stripes, dark stripes, light stripes, dark stripe, light stripes. When you zoom out and you look at a sarcomere, what creates those stripes are A bands and I bands. An A band gives you a dark stripe. So an A band starts from one end of a thick filament to the other end of a thick filament. And that thick filament, because it's thicker than a thin filament, creates that dark stripe that's associated with an A band. An I band gives you a light stripe. So an I band is from an end of a thick filament, and it crosses the Z line. So let's pretend like we're crossing the Z line, and we're starting on another sarcomere. And we are crossing that Z line over here. So the I band is from this sarcomere. Let's pretend like that's another one to this one. So from one end of a thick filament to the beginning of another thick filament is an I band, and that creates the lightness. So what that looks like over here. Okay, so number 13 here is the A band. And then the I band is going to be number 12. So you see how it crosses that Z? That's what creates our light band, our I band, I light, and A band dark right there. Okay. In these two, and then H is just that space between the M line and the thin filament. I don't know if I said what H was. Okay. So this is a relaxed state. So the sarcomere is relaxed. Once it contracts, the Z line is going to move inward. So this Z line moves inward, this Z line moves inward towards the M line. What happens is the space, the age zone right there, gets narrower, gets more narrow after contraction, and the I band gets more narrow after contraction. Everything else stays the same length. So the H and the I get more narrow during contraction, but nothing changes with the length of the filament. All that happens is that space gets closer, everything gets closer together, but the length of the actual filaments do not change. Okay, so the length of the proteins don't change, they simply move closer together. The gaps disappear during contraction. So the distance for H and the distance for I get closer together. H gets shorter, I gets shorter. Or I should say, H and I get more narrow. High gets more narrow during contraction, but the fibers themselves don't change length. They simply just move closer together. Okay. So this is a good worksheet to print out and work through to kind of study for this. We will go over it together though, so you know you have the right answers. So number 12, right here, that's the I band. So that's the light band, I, light, and it is bisected by the Z line between the two sarcomeres, right? It's a thin filament, it's actin only. And then we have the A band, which is a dark band, thick filament, and it spans the entire length of the myosin right there. From end of thick filament to end of thick filament, that's the A band. Number 14 is the H zone, so that's the central region of the A band, and it's the space between the M line and the thin filament. It's the H zone. See, number 15, that's the Z line. So from Z line to Z line is one sarcomere. That's question 21 is a sarcomere. Number 16, that's just called elastic filament. Number 17, myofilament. So thick band, thin band, thick band, thin band. That's just they're both myofilaments, myosin and actin. Myosin is thick, actin is thin. M line, so number 18 right here, that's the M line. Connects the thick filaments, center of the sarcomere. It anchors the thick filaments, the thick myosin filaments. See, number 19, that's the thin filament, this red. Thin filament, actin. It slides over myosin to shorten the sarcomere during muscle contraction, like we saw earlier, and that's also what you saw in the video that you watched that's embedded into this one. And then the other two YouTube videos also showed it. Number 20, that's a thick filament, so myosin. And that one forms a cross bridge with actin during contraction. It's a motor protein, so basically that converts chemical energy from ATP to do work or movement inside cells. And then the sarcomere, number 21, from Z line to Z line. But yeah, use this, study it. You will have to know it. All right, myofilament. So this is where it gets a little bit more difficult, just in terms of all the terminology that you have to learn for this. But I'm going to try and keep it as simple as possible, right? So myofilaments. So thin filaments are made up of actin, which are these blue pearls in this myofilament right here, or in this myofiber right here. So actin are the blue pearls. Troponin is a 3-bead complex that's in these purple ovals. And then tropomyosin is this yellow-brown string in the middle. Here in this example, troponin are these blue 3-bead complexes. The yellow pearls are the actin, and the tropomyosin is this blue string that goes through the middle. So that's what it looks like in both of these models. The thick filament is made of myosin. So that's this red fiber. It has flexible heads with binding sites to attach to actins. So the heads look like little golf clubs, right? Those are little heads for the binding sites. And the heads are turned away to face out, and then they spiral up the side of the thick filament. So there is a place on actin for the myosin heads to attach. Right now, the tropomyosin the string is covering up the attachment points for the myosin heads when the muscle's relaxed. So the troponin is the control mechanism for getting the tropomyosin to move out of the way. So the troponin beads here, one is going to attach troponin to actin to make sure it stays attached to the thin filament. Another one is for calcium molecule. And then another one is attached to the tropomyosin. So calcium comes in, attaches to one of the troponins that's meant for it to attach to, and it changes actually the shape of that troponin, and it helps move that troponin out of the way. As it does that, it pulls the tropomyosin with it. And then the attachment point on myosin for the actin beads will be exposed. You'll be able to see it to where myosin can attach because the tropomyosin string has been pulled out of the way, right? Then we have energy. And then the heads go from being flipped out to flipped inward. So see how they're flipped out? They would flip inward and drag everything in towards the middle, right here. So remember, the lengths don't change, they just get closer together until the muscle contracts as far as it needs to. Okay, this is exactly what we just talked about, but we're gonna go over it again with words instead of pictures. So a nerve stimulates, creates an action potential, and increases calcium in the cell. Calcium then binds to troponin. Tropomyosin then gets out of the way and allows actin to attach to myosin head. Then energy is going to change. ADP and P release causes myosin head to flip inward, and then ATP attaches to myosin and causes the heads to flip out again to relax. And then the enzyme ATPase comes in, breaks ATP down to ADP and P, and then we're ready for the next contraction. So basically, you have stimulation, an action potential, an increase in calcium, binding to troponin. You're moving away from the binding site, heads are going to flip. When the heads flip back out, relaxation happens. And this is a rowboat cross bridge analogy. So resting, action potential comes in, myosin actin binding site. So resting, action potential, catch. Then we drive, so power strokes. Now we've got the energy, ATP binding and acting, myosin release, and recover, resting right there. Okay. So this is pretty intuitive. The more muscle fibers that you stimulate, the stronger the force. The bigger the muscle fibers, the stronger the force. Big biceps are always going to be stronger than small ones. The more frequent a stimulation, the stronger the force. And then if you're at 50% contraction at the start, so if your muscle's already partially contracted, primed, ready to go, ready to lift something, you're already stronger than you would have been if you were at a relaxed state trying to lift something. So also, we build muscle by tearing it down, right? Basically, stimulation from a workout is going to put tension or damage on a muscle. Then when we're resting, those damaged fibers are going to fuse together with help of satellite cells to help repair that muscle. As that's happening, it's actually building muscle back thicker and stronger, so synthesis. And over time, we're at the growth stage with training and recovery to increase overall muscle mass. That's all called hypertrophy. So I did want to mention that. And then because we've been talking so much about ATP, so what happens during muscle contraction, just quickly on different types of respiration and energy yield. So I know that most of you have heard about slow twitch and fast twitch muscle fibers. So we're going to talk about aerobic respiration just shortly and anaerobic. So aerobic respiration is what energy is being produced with available oxygen. So we have more energy yield here in terms of long-term and sustained energy. These are our slow twitch muscle fibers. They're pretty resistant to fatigue, and they're characterized with jogging, marathon runners, again, sustained long-term energy. Well, when our muscles use up all of that oxygen, and we're using more oxygen than what's being supplied, anaerobic respiration kicks in, right? So energy yield with aerobic was 36 ATP. Anaerobic is only 2 ATP. Again, we're not using oxygen here. With anaerobic, we get short bursts of energy. These are our fast twitch muscle fibers. So they are powerful, but they fatigue really, really quickly because we don't have as much energy yield, right? So I'll just read through this. Anaerobic respiration occurs when oxygen is not available. So our muscles have used up oxygen faster than what is being supplied. So our muscle cells then switch from aerobic to anaerobic so we can keep producing at least some ATP, right? So we can keep some energy sustained. Anaerobic allows for short bursts of energy, but it causes muscle fatigue and soreness due to lactic acid buildup in the muscles. So once lactic acid accumulates in the muscles, remember our pH scale, we're getting more acidic. That's what causes that burning sensation that you feel with muscle soreness and fatigue. And that's because we have used up all of our oxygen and we have gone into anaerobic respiration. And that's why we are sore and fatigued. So that's just a little bit on that. And then just some disorders that can happen with the muscular system to end this lecture on. So cramps, right? Painful muscle spasm caused by rapid fire of motor neurons. We know what cramping is. Crush syndrome happens. Shock-like state following massive crushing of muscles. So this is potentially fatal. High fever. This is especially fatal because myoglobin can go into the urine and cause a super big fever, and everything messes up, and et cetera, et cetera. You see crush syndrome in a lot of doctor hospital type shows. Delayed onset muscle soreness, pain, stiffness, tenderness, felt from several hours to a day after strenuous exercise. So this is what we were talking about in terms of breaking down muscle to build it back up, micro trauma, and lactic acid buildup. Disuse atrophy. This happens in people that are bedridden. So muscle strength can be lost at a rate of 3% per day of bed rest. And this can happen for any sort of reason, whether post-surgery or something happened to make you bedridden. Fibromyalgia is chronic pain, and actually there's no known cause as of right now. We just know that it happens in women about twice as often as it happens in men, early onset between ages 30 and 50. And then myositis can happen from autoimmune diseases causing muscular inflammation and weakness. Oh, and then contracture, that first one, abnormal muscle shortening not caused by nervous stimulation. So failure of the calcium pump to remove calcium from the sarcoplasm or from contraction of scar tissue, as in burn patients. Pretty interesting. Okay, well, that's it. That's it for this lecture.